Hello everyone. Today we're going to talk about principles of protein structures and protein folding. So as you must have learned from biochemistry class, so there is a hierarchy in protein structures. So first, the proteins are chain molecules. Proteins are chain molecules, and their basic building blocks are amino acids. So we call primary structure. So what we call primary structure is just a sequence of just a chain of amino acids. So the primary structure is about the order of chain. So how amino acids are connected, starting from alanine and leucine or whatever, phenylalanine and something like that. So how they are just ordered. So the primary structure is a one-dimensional. So it's just the order of 20 amino acids. And after that, so there is a structure called secondary structure. Secondary structure. So the secondary structure means that locally, so there are many, repeating structures in protein's 3D structure. So for example, so the substructure called alpha helix and substructure called beta sheets are frequently observed in protein structures. So these two are the main local building blocks. Main local building blocks of protein structures. And based on these secondary structures, so the structure called tertiary structures are generated. So the tertiary structure means that how proteins are three-dimensionally ordered. So how they are folded. How are they folded? So the tertiary structure is the complete 3D structure of the whole protein chain. And after that, there's also a structure called quaternary structure. So the quaternary structure is about how individual proteins are assembled together. So here, different colors mean different individual protein chains. So the quaternary structure is a protein assembly. It's about protein assembly. So individual protein chains are aggregated, assembled together. So that's called quaternary structure. So as we have talked before, so the amino acids are the basic building blocks of protein structure. And amino acids, so the basic structure of amino acid is shown on the left side. So as you can see here, these parts are called backbone, and they are the same for all amino acids. They are same for all amino acids, and only this R group, this functional group, is substituted, is changed. And in natural amino acids, so in nature, only 20 amino acids are used. Oops, I'm sorry. So the only natural proteins consist of only 20 amino acids. So as you know, as you learned from the organic chemistry class, so this R group can be anything. This can be arbitrary. But in nature, only actually 99.9% of organisms use only 20 amino acids. So there are very rarely, there are some microbiomes which use very rare amino acids, but most of the organisms we know use only 20 amino acids, which are shown on the right side. And so some are negative and some are positive, and some are uncharged polar groups, and some have non-polar groups. And this R group is called side chain. This R group is called side chain. So based on the property of this side chain, so it determines the whole physical property of amino acid is determined. So this is the table. This is the structure of 20 amino acids. So for example, the simplest one is glycine. So the glycine has H at R group at R position, and alanine has a CH3, so this would be a non-polar amino acid, and valine has this structure, leucine, isoleucine, and methionine, phenylalanine, tryptophan, and so on. And also some amino acids are polar. Polar means they have dipole moment. In their R group. For example, serine has OH at the end of its R group, and threonine has OH here. Cysteine has SH, tyrosine has OH, and asparagine has NH2 and O, and glutamine has NH2 and O, but it has two CH2 groups in between. So these R groups, these side chains have polar groups at the end, polar atoms at the end, so their net dipole moment is not zero. So these non-polar groups also has non-zero dipole moments, but their dipole moments are almost very close to zero. But this one has a polar dipole moments, as you know. These OH groups or SN groups can form hydrogen bond, right? And some R groups are actually charged at pH 7.4 actually. For example, aspartate and glutamate have carboxyl groups at the end. But as you know, these carboxyl groups are generally charged at pH 7, right? So our body, most of living organisms have pH 7 point something at their normal condition. So they are generally so these carboxyl groups are generally negatively charged at ambient condition at around pH 7. And lysine, arginine, and histidines are generally positively charged at around pH 7.4. So as you see, so this lysine has, NH3 plus group at the end, and this arginine has NH2 and NH2 plus group at the end, and this histidine has nitrogen containing ring, 5-membered ring, and one of these nitrogens can have protons, and so that they are positively charged. So then how these amino acids are determined? So actually, in the nucleus of cell, so let's say that this is a cell and this is a nucleus. And inside a nucleus, there is a DNA. There is a DNA double helix, right? DNA. And DNA consists of 4 nucleic acids, right? And based on the combination of these 4 nucleic acids, actually these nucleic acids are translated to amino acids. And it is known that 3 nucleic acids are translated to 1 amino acid. And these 3 combinations of nucleic acids are called codon. And table on the right side shows the codon table. So for example, so this is the RNA codon table. Actually, in RNA, they use uracil, but in DNA, uracil is substituted to a thymine, right? So this is in DNA, this is T. So here, as you see, this column corresponds to the first position, and this row corresponds to the second position, and this column corresponds to the third position. For example, so in DNA, so U, if U amino acids are ordered in their DNA like this, then this U is translated to phenylalanine. So the U, the first one is U, and second one is U, and third one is U. Then it is translated to phenylalanine. If RNA has a sequence of CUA, then it starts with C, and the second one is a U, and the third one is A, then it is translated to leucine. And if RNA has a sequence of GAC, for example, then it is translated to aspartic acid, right? So that therefore, so if you think about the combinations, then if you think about the position 1, position 2, and position 3, then each position can have 4 possibilities, right? 4 possibility and 4 possibility. So the all possible combinations are sum up to 4 to the 3rd order, so which corresponds to 64 combinations, right? But if you remember that, so there are only 20 amino acids, then which means that, there are 64 combinations, but they are translated to 20 amino acids, right? So which means that some codons are translated to the same. So some codons are translated to the same amino acid, but some codons are translated to a very unique amino acid. So for example, so if you see that these 6 combinations are actually translated to leucine, and these 4 combinations are translated to proline, and these 6 combinations are translated to serine, and something like that. And some are translated to stop codons, and so on. So why do some codons are translated to the same protein, but for example, in case of methionine, only one codon is translated to methionine. So why are they so different? So it is still not understood fully. So it is one thing we should understand in the future. But the fact we know now is that so some codons are translated to the same amino acid, but some codon is translated to only one amino acid, and so on. So based on this codon table, so the DNA information is transcribed to the RNA, and RNA information is translated to a protein, becomes protein. Okay. So actually, this is the movie about the protein translation. So this should be a movie, but so here so since I'm giving a lecture on PDF file, so I'm going to provide you the link of this movie file. But I recommend you to watch this movie. So since it is very well made. So based on this codon table, so this codon is translated to protein, and in protein, as I mentioned, so protein is a chain of amino acids, and how they are connected. So if you see these amino acids, so let's say that there are these 4 amino acids, and they are connected through this peptide bond. If you see, so at the end of one amino acid, so there is a COO group, and at the another end of this amino acid, they have NH3 group. So if they react, then OH2 molecule leaves. So the OH2, water molecule is formed and it leaves. And the rest of the so let's say that so this one and these ones are combined, and the rest of the group forms covalent bond. Okay, so they form bond something called a peptide bond. So there is a CO2, COO, and N and NH. So they form a single covalent bond, and this is called peptide bond. So through these peptide bonds, so amino acids form a long chain. So this is how amino acids are formed a long protein. So this is called peptide chain. So once they form a chain and they are folded to a compact structure. They are folded to a compact structure. And based on something called Anfinsen's principle. So in this slide, we're going to talk about Anfinsen's principle, Anfinsen's experiment. So in 1972, so the Anfinsen found out that once protein, if there is a stable protein, and protein can be denatured. Denatured means that they become flexible. They are unfolded. So denatured means unfolded. They become unfolded. And so for example, once there is a normal protein, and if you increase pH or decrease pH, and if you increase temperature, and if you increase ionic structure, and if you increase solubility, and they become denatured. Denatured means, as I said, they become unfolded. But once these, external conditions back to normal condition, they are folded again. But they are always refolded to its native structure. So they are always refolded to its original structure. So that's what Anfinsen found out in 1972. So Anfinsen did this several times, but they are always, unfold. So they start from the stable folded structure. So this is the native structure, okay? So the native means that they stay in this structure. So once they at their stable temperature and stable condition. But from this native structure, if you change environment, such as, protein pH becomes very high and very low, or when denaturant is added, such as, urea. So if you add urea to a protein solution, then proteins become, unfolded. But once urea is removed and they go back to their original state. This is what Anfinsen found out, and this is something called, Anfinsen's principle. So the everyday example of protein denaturation is an egg fry. So if you break an egg on a frying pan, and they are at first they are transparent, right? But if you heat them to a high temperature, then they become, denatured. But for eggs, unfortunately, so they are just full of eggs. So once they are denatured and they are aggregated and they are entangled to each other, then they become like this, right? So this is actually the result of unfolding plus aggregation or entanglement. Okay, so if you heat protein and they become, they change their structure. And urea is a molecule like this, right? And just the one trivia is that the urea is the first organic molecules, synthesized from inorganic compounds. Then why are proteins affected? So one thing we should talk about is that why are proteins affected by pH? So as we mentioned, there are 5 amino acids. There are 5 amino acids which are assumed to be charged at ambient pH, at pH 7.4, but actually, their charge states can be changed by environmental pH. So for example, so for glutamate, they have a carboxyl group at the end, but at low pH, these carboxyl groups likes to be connected with protein. Likes to have proton at the end. So they want to be at this form. And also this end, let's say that we have a glutamine monomer in a solution. But if we add more and more OH minus group, which means that they are titrated, then this carboxyl group loses its proton first. And if we titrate more, if we add basic species more and more, then this proton will leave and this becomes negatively charged and this one becomes negatively charged. And if we add more and more OH minus, then proton at this NH3 group will also leave, and this becomes some this one has a -2 charge. So the protonation state of these 5 amino acids are affected by pH and their pKa, and their ratio between HA and A- can be calculated by this Henderson-Hasselbalch equation, as you have learned from the biochemistry class, right? So pH equals pKa plus log A- over HA, right? So this is why pH affects the structure and stability or the behavior of proteins.